This video is going to be about the prokaryotic cells. So, prokaryotic cells, or the prokaryotic organisms, are divided into two major groups. We got the eubacteria, which includes all bacteria of medical importance. And then we have the archaebacteria, which is a collection of evolutionary distinct organisms. Here are some characteristics of a prokaryotic cell. So their chromosomes are usually single, circular in shape, plus they have plasmids. They have no nuclear envelope or nucleoli. Also, they do not have any membrane-bound organelles. They have cell walls. Some are complex. They also have a plasma membrane, but they lack the sterols in there. And their average size is about 0.2 to 2 millimeters. They replicate through binary fission, and they also have no histones. As I said, the cell wall is a little complex. That is because the cell walls are categorized according to their staining characteristics. So they will either be gram-positive or they will be gram-negative. The gram-positive cell wall. I'm going to use a flowering pot to demonstrate how this looks like. So we're going to start with the bottom layer. And so let's draw the plasma membrane. We've got the dotted line. And then we got some integral proteins that are going to be embedded into the plasma membranes. The plasma membrane is similar, but it does lack the sterols, so you have to remember that. And then above that, we got the peptidoglycan. Now, the peptidoglycan is a very thick protective layer, could also be called a murin layer. This layer consists of glycan chains of alternating N-acetylglycosamine, NAG, and N-acetylmuramic acid, NAM. So these two chains alternate and they make up the peptidoglycan layer. Now within this layer, we also have the teichoic acid. So this I'm going to draw with the red lines. So the teichoic acid, it is anchored to the murin. And then we have lipoteichoic acid, which I'm going to draw in the color white. The lipoteichoic acid is actually anchored to the plasma membrane. So it goes all the way down to the plasma membrane. So as you can see, it is longer. Now, these are the characteristics that are unique to the gram-positive cell wall. Yeah. Gram-negative cell wall. I'm going to use a CD to describe this because it's a bit more complex and it also is composed of two membranes. So we got the inner cytoplasmic membrane. Outside this, the large empty space where I'm going to draw these squiggly lines, that is a periplasmic space. This space is much bigger than that of in gram-positive. That's because it contains degradative enzymes and also has some transport proteins. Outside this space, we got the small layer, which is called the peptidoglycan layer. Again, this layer is much smaller than one in gram-positive, and because of this, the cells are consequently more susceptible to physical damage. Then we got the outer membrane. The outer membrane is distinguished by the presence of embedded lipopolysaccharides, LPS. We got some proteins, and we got some phospholipids, which are in the outer membrane of a gram-negative cell. So here is a side-by-side comparison of the gram-positive and the gram-negative cell wall. This was just meant to be a short, quick video to just let you know what the difference between the two types are and what are prokaryotic cells. I hope you guys like it. This was my first time making a video. So yeah, I hope you guys learned something.